Hello, everyone. Welcome and good afternoon. My name is Benjamin Quinn, and on behalf of Harvard Bookstore, the University Division of Science, and the Harvard Library, I'm so excited to welcome you to today's virtual event with Paul Nurse, presenting his new book, What is Life? 5 Great Ideas in Biology, with a special introduction by Harvard's own Andrew Murray. I hope you're all well and safe today. Thank you for joining us virtually. Today's event is the latest installment in our Harvard Science Book Talk series, which works to bring the authors of recently published science literature to our Cambridge community and beyond. Coming up in the series this Friday, April 2nd, we'll host Nobel laureate Steven Weinberg and Harvard's Andrew Strominger for discussion of their work and their books, The First 3 Minutes, A Modern View of the Origin of the Universe, and Third Thoughts, The Universe We Still Don't Know. To learn more about this and our other upcoming virtual events, you can visit harvard.com and sign up for our email newsletter, or check out the page harvard.com/science for more info. We also have a Science Research Public Lectures YouTube channel where you can view previous talks you might have missed. This afternoon's discussion will conclude with some time for your questions. If you have a question for our authors at any time during the talk today, click on the Q&A button at the bottom of the screen and you will get through as many as time allows. In the chat, I'll be posting a link to purchase What is Life on harvard.com, as well as a link to donate in support of this series and our store. Your purchases and financial contributions make events like tonight's possible and help ensure the future of a landmark independent bookstore. Thank you to our partners at Harvard University, and thank you all for showing up and tuning in support of authors, publishers, indie bookselling, and especially for science, because it really, really matters. And finally, as you may have experienced in virtual gatherings this past year, technical issues may arise. If they do, we'll do our absolute best to resolve them as quickly as possible. Thank you for your patience and your understanding. So now I'm excited to turn things over to Andrew Murray, Director of the Rollin Institute and the Herschel Smith Professor of Molecular Genetics at Harvard University. The digital podium is all yours, Andrew.
Thank you so much. Oops. You bring the video back. Thank you so much. Life is full of little surprises. I was looking at my email the other day and I saw an email from a colleague, Melissa Franklin, saying that Paul Nurse was coming to talk at the Harvard bookstore and would I kindly introduce him. And it's an extraordinary pleasure to introduce someone who is a hero of mine, both for their humanity and for their abilities as a scientist. Paul was born under rather unusual circumstances in Norwich, a city in the east of England, and you can read about the unusual circumstances in his book. And he grew up, he became interested in science. He attended the University of Birmingham, and he then went to do a PhD at the University of East Anglia. And while he was there, working on a machine that was old and tiresome and troublesome, he would read papers at night and look for problems to be interested in after his PhD. And he fell upon the problem of how cells grow and divide. And so Paul then went off to Edinburgh, where he isolated the first mutants that gave us a clue as to how cells know how big to grow before they can divide. He pursued that research from Edinburgh to Sussex to London to Oxford, back to London again. And he produced such monumental advances that he, along with Lee Hartwell and Tim Hunt, won the Nobel Prize for Medicine or Physiology in 2001. So he's been an extraordinary, thoughtful, and inventive scientist. He's also been a leader of science. He directed the Imperial Cancer Research Fund, Cancer Research UK. He's been president of the Rockefeller. He's been the head of the Royal Society, and now he's the head of the Crick Institute in London. But the thing that I most admire about Paul is that his extraordinary intellect is melded to a remarkable sense of humanity. He's one of the kindest, funniest, nicest people I know, and it's an extraordinary pleasure to introduce him to talk about his book, What is Life? Thank you, Paul.
Well, thank you, Andrew. that was a very nice introduction, and I'm rather don't quite know what to say, actually. Maybe there's some truth in some of it. I'm going to share my screen because I'm going to show a few slides with respect to my book, which I now will. There we are. Okay. Now, what am I going to talk about today? I'm going to talk about the title of the book that's just been published in the US called What is Life? It's one of the fundamental questions in biology. Some would say the most fundamental question, but it's very much easier to ask than it is to answer. If you look around us, there's an extraordinary diversity of life on our planet. There's bacteria, fungi, plants, animals, including ourselves, human beings. These all maintain themselves, they grow, they self-organize, they reproduce. But what is it that links all these different life forms together? What do they share? I think it was a butterfly that first started me thinking about questions like this. It was early spring. I was perhaps 12 or 13 years of old. I was sitting in the garden in northwest London, where I'd brought up after I moved down there from Norwich, when a quivering yellow butterfly, this is it, a yellow brimstone, flew over the fence. It turned, it hovered, it briefly settled, a shadow disturbed it, it took flight again and disappeared over the opposite fence. That intricate, perfectly formed yellow butterfly made me think. It was both utterly different to me and yet somehow familiar too. Like me, it was so obviously alive. It could move, it could sense, it could respond. It seemed so full of purpose. I found myself wondering, what does it really mean to be alive? In short, what is life? Now, this question has been asked by others and been asked for years by others. For example, this is the title page of a book called What is Life? Same title, I stole it from him, written in 1944 by the famous physicist Erwin Schrödinger. His book was influential, including for me when I first read it now 50 years ago. Now, I'm going to ask the same question that Schrödinger asked, but I'm going to do it in a way to consider what I think are 5 important ideas of biology. And there's a bit of history of ideas here because I'm going to describe where those ideas come from, explain what they are in a way that anybody can understand, at least I hope so. But then at the end of my talk, I'll pull them together to develop principles that I think are fundamental to understanding life. Now, the first of these ideas is the concept of the cell, biology's atom. Now, the cell is the basic structural and functional unit of life. It really is life's atom. And they were discovered by Robert Hooke in 1665. He was using a microscope, like the one shown on here, and the image in black and white is one that he drew. And what he saw, after he cut a slither of a cork with a razor, looked under this microscope and he saw neat arrays of boxes. He called these boxes cells after the Latin word cella for small cubicle, and it's shown here in his drawing published in a book called Micrographia. I put here also a modern scanning EM photograph of the same material. Now, a few years later, a Dutch draper, Anthony van Leeuwenhoek, who worked in Delft in the Netherlands, scraped between his teeth and discovered single-celled life, bacteria. He sent this drawing, it's one of 1,000 he sent, to the Royal Society in London that had been founded about 20, 30 years before. And it had, this picture had a charming image, at least I think it's charming, of a bacterium doing a loop-the-loop under the microscope. You see it here shown in Fig B to C to D. He noted he was rather disturbed by this study of a microorganism living in his teeth because he was very proud of his dental hygiene. Now, he was a close neighbor. He lived in Delft in the Netherlands, and he was a close neighbor to the painter Vermeer, who painted two pictures of a scientist. This is one of them. It's in the Louvre. And I like to think that this might be Leeuwenhoek. It's called either the astronomer or the geographer. I like to think this is possibly a Leeuwenhoek, but I have to admit to you, there's no good evidence for this speculation. Although when Vermeer died, Leeuwenhoek was the executor of his will, so there may have been some connection. Now, this was in the 1680s, 1690s, and over the next 2 centuries, it became clear that all living organisms, not just cork derived from a plant, all living organisms consist either of a single cell or a collection of cells. And this conclusion was summarized by this stern-looking German zoologist in the 1830s, Theodor Schwann, who in 1839 wrote in German, but it's translated here, all organisms are composed of essentially like parts, namely of cells. Now, as well as cells being the basic structural unit of life, they are also the basic functional unit of life. And this was expressed by another German scientist, the pathologist Rudolf Virchow, who in 1858 wrote, and I put it here, that the cell is a vital unit bearing the complete characteristics of life. And that it really defines that a cell is the functional, the basic functional unit of life. He also said that all cells come from cells, omnicellular acellular. And this simply means that cells arise through the division of pre-existing cells. They don't arise in the corner of a cell, or they don't arise spontaneously. So cells are really interesting, the basic unit of life. Now, if I have failed to convince you that they're interesting, let me just show you this picture. This is a mammalian egg being fertilized. You see all these sperm trying to fertilize this egg. And all I need to do is remind every one of you that you were all once a single cell, just like this egg. And to be honest, if you're no longer interested in cells, then maybe it's now time to switch off the webinar. Now, cells are considered to be the simplest entities which unambiguously, and you'll see why I say that towards the end of my talk, unambiguously exhibit the characteristics of life. And that means understanding cells is key to understanding life. So they will figure prominently in what I have to say. Now, here's a picture, a modern EM picture of a cell. You can see that a cell is a bounded physical entity. It's separated from their environment by a semi-permeable lipid or fatty membrane that separates them from their surroundings, but allows communication with those surroundings. Now, this separation allows order to be built up inside cells at the expense of increasing disorder outside cells. This sounds like physics because it is physics. And I say it because in this way, life does not contravene the second law of thermodynamics, which states that the universe as a whole moves towards disorder. This is often noted by physicists thinking about life, including Schrödinger, who, to go back to his 1944 book, he had a couple of quotes. He was astonished, if you look at the top quote there, that life could escape the decay into atomic chaos. And this escape really fascinated him because he was astonished that life could do that. And it led him to propose that they had to have a code script that was inherited from one generation to the next. And these two quotes from Schrödinger's 1944 book introduced a second idea that I want to share with you. And that idea is the gene which can explain the mysterious phenomenon of inheritance. Now, this is the idea, of course, that the gene is the basis of heredity. And it had its origins in the work of the Augustinian monk Gregor Mendel, who was abbot of Brunno Monastery. And he published his work in 1865, in the middle of the 19th century. And here he is in his monk's garb. Now, his monastery actually was a sort of scientific research institute, really. And he had a greenhouse and gardens. He had a telescope where he made a map of the moon, meteorology where he would follow the weather. And he tackled the problem of inheritance by carrying out crosses with plants, which was quite popular at the time. There were quite a number of scientists, particularly in continental Europe, who were doing that. He crossed pea plants that had different characteristics and outlined the outcomes of those crosses. And he had a big greenhouse, as I said, and also a garden. And I found this picture of his garden. This is from 1981, when I first visited his monastery, right in the middle of the Cold War. It was certainly not easy, in fact, to get there. Now, what Mendel did was to cross peas of varied characteristics, including plants of different heights, different flower colors, different seed shapes. And he chose characteristics that he could very clearly distinguish one from another. So he'd have a clear way of defining the outcomes. And I'll give you a picture here showing some of the sorts of pea phenotypes, characteristics that these types of plants can exhibit. Now, when he did these crosses, he famously observed rather simple ratios in the outcome of these crosses, the progeny produced by these crosses. And ratios would emerge like 3:1, which many of you are probably familiar with, some from school, for example. And this eventually led to what we now call Mendelism. It wasn't Mendel himself. It actually needed a little bit more work after he'd conceived the idea, and he was basically forgotten for 30 or 40 years. But the idea of Mendelism is that heredity is based on indivisible particles or factors that are inherited from generation to generation through the germ cells. That is the pollen and ovules in plants and sperm and eggs in animals. These factors, after Mendel, 50 years later, were found to be linked to the chromosomes that were discovered to be separating in dividing cells, as shown here in this late 19th century drawing of onion root tip cells. Now, these Mendelian hereditary factors are what we now call genes. In the 1940s, Oswald Avery in Rockefeller University, New York, which Andrew mentioned, I worked for 7 or 8 years. And in fact, Andrew did a sabbatical there. I was president there. What Oswald Avery in the 1940s and his group demonstrated was that genes were made of deoxyribonucleic acid, or DNA. And here is a scheme of DNA coming out of the nucleus where it's found the chromosome, unraveling the chromosome, and there you can see DNA. Now, this is the famous double helix, the structure of which was established 10 years later in work that occurred in London, experimental work by Rosalind Franklin, Ryan Gosling, and Maurice Wilkins, and then the theoretical interpretation of that work in Cambridge by Francis Crick and Jim Watson. Crick and Watson famously argued that the double helical structure of DNA could explain heredity through the linear sequence of bases making up DNA and the pairing between complementing bases, adenine pairing with thymine and cytosine pairing with guanine. Now, this is shown here. The double helix is like a twisted ladder. Imagine a ladder with rungs going across, one half of which will be one base, adenine, say, the other half of the rung being thymine, same with cytosine and guanine. The rungs of the ladder are made up of the links between these complementing bases. And when these links are split, two templates are formed, which are used to make two absolutely identical helices. And this explains how DNA is copied during cell reproduction, and it accounts for heredity. It, of course, also explains Schrodinger's code script and the permanence of the gene between generations, because DNA acts as a code script, which is precisely copied every time a cell divides. And the sequence of bases encoding that genetic information is kept nice and stable and chemically protected in the middle of the DNA polymer double helix. Now, Crick went on to propose what he called the central dogma, explaining how DNA encodes the structure of proteins, which I'm going to talk about a little later in this talk. The central dogma describes how the sequence of bases making up a gene is copied from the DNA to ribonucleic acid or RNA, and this acts as a messenger, the RNA, between the DNA located in the nucleus, where the chromosomes are, and the cytoplasm, where the proteins are made. And the sequence of the messenger RNA, determined by the sequence of the DNA, determines the structure of proteins. And others showed, working with fungi actually, that there's one gene for each protein. These discoveries were made around the second half of the 20th century, and they laid the foundations for the revolution we now call molecular biology. They are critical for understanding life. And central to this is the genetic code. Heredity is written in this 4-letter code made of the 4 bases, A, T, C, G, and the information is stored in a linear form. Now, this way of storing information is the same in principle as sentences in a book, the words I'm speaking in this talk, or for that matter, bytes in a computer. But what it means, of course, is that the linear digital storage of information was invented by life over 3 billion years before the computer age. Now, the nature and function of the gene introduces the next 2 ideas I want to explain to you: life as chemistry and life as information. And these are important ideas, and I'm going to start with life as chemistry. The functioning of living organisms is based on chemical activity and physical forces. The origins of the idea can be traced back to French chemists. After initial work of Lavoisier, the father of chemistry, who unfortunately lost his head in the French Revolution, by the way, it was taken up by Louis Pasteur some 50, 70 years later, who carried out experiments on sugar beet fermentation in the north of France, which led to the concept that life is chemistry. And here's Pasteur, Louis Pasteur, heroically doing experiments in his laboratory. He was analyzing in the north of France the fermentation of sugar beet, which was carried out by industrialists to make alcohol. And at the request of these industrialists, he was asked to investigate why these fermentations sometimes failed and didn't make alcohol, but made acid instead. He demonstrated that successful production of alcohol required the activity of a living organism, a single-celled one, yeast. He proposed that the yeast cell produced substances that made alcohol from the sugar derived from the sugar beet. And if yeast was not there, but bacteria instead, then the fermentation made acid and not alcohol. Now, that solved the problem for the industrialists, but it wasn't enough for Louis Pasteur, because he made a general conclusion. He concluded that fermentation, what he had been studying, was a physiological act yielding products for the cell, and concluded that chemical reactions are expression of the life of the cell. And in other words, that life is chemistry. But I tell you, it's a very elaborate, a very special chemistry, and it's one that's based on carbon polymers. Now, we've already met carbon polymers that make up the nucleic acids, DNA and RNA, and now we need to consider the carbon polymers that make up proteins. The substances from yeast that Pasteur postulated made the alcohol are proteins that act as enzymes, enzymes which catalyze chemical reactions. Proteins are polymers of amino acids based mainly on carbon and nitrogen. The carbon atoms form the backbone to a polymer like the one shown here, and also forms links to side chains on the amino acids, which are marked by the R identified yellow circles here. The structure of a polymer is shown here, and schematized here, which shows that the different amino acid chains are chemically different. So these side chains are very, very different looking, as you can see: lysine, leucine, two leucines, tyrosine, aspartic acid. Some are positively charged, like lysine, some negatively charged, like aspartic acid, some repel water, some are big, some are small. And these are just examples of the 20 different amino acids that make up proteins. And as a result of this, there is a really wide variety of chemistry that can produce many elaborate structures which carry out the varied chemical reactions. And this contrasts with DNA, which is stable, it's not chemically diverse, and in fact is chemically rather dull. Now, the chains of these amino acids make the protein polymers that fold up in many ways to make complex molecular structures. And I show here a protein chain of quite a lot of amino acids, and a three-dimensional molecular structure derived from that one-dimensional chain. And these varied structures, combined with the varied chemistries of the 20 different amino acids, accounts for the wide diversity of chemical reactions that enzymes carry out. You should think of enzymes and other proteins as chemical machines. These machines produce a huge range of metabolic chemicals required for living cells, living organisms to function. They make new molecules, they break them down, they recycle them, they generate molecular assemblies which synthesize these larger polymers of nucleic acid and protein and lipid and carbohydrate. They also do physical work. They make motors which carry cargoes around on protein-based trackways. They act in hybrid ways, combining chemistry and physics, capturing energy from sunlight or breaking down sugars to produce energy-rich chemicals that drive the activities in cells. Now, all this activity means there's thousands of chemical reactions being carried out simultaneously, close to each other in living cells. The small examples of these are shown as dots in this map of the central metabolism of a cell, part of the chemistry of life. Now, because these reactions require different chemical conditions, they have to be separated from each other. This is achieved by compartmentation, brought about at different levels by the surfaces and folds of the enzymes, by complexes of several enzymes acting together, by membrane-bounded organelles, which are visible here. This is the picture you saw before, and schematized here. Now, the reason I wanted to show you a cell again is because a different way of thinking about cells is that they are compartments which are formed by many different chemical microenvironments. And it's this compartmentation from the level of a single protein to an organelle which allows the hugely elaborate chemistry of life to take place. But for all of this to work, this chemistry must work together as a whole. It has to be organized together. It requires communication between these different chemical microenvironments and compartments. Communication is the transfer of information. And that leads to the next idea, life as information, centered on complex systems, on control, on purpose. Now, life is complex. We know that. It's a complex system. Curiously, it was first argued, at least the first example I could really find, by the philosopher Immanuel Kant. He wrote a book on moral philosophy and had a speculation about the nature of life. and the operations of the complex systems of life are based on the management of information. I want to give you two examples to illustrate this. The first is to go back to the structure of DNA. Now, this structure of DNA, it's iconic, of course, it's beautiful, of course, but its real beauty is not in this structure we see here, but in the fact that it only makes sense biologically when it is recognized to be a digital information storage device, one written, as I've already explained, in the linear script of 4 letters. And this linear polymer is ideal for storing information, with the encoding letter bases turned inside the helix, protected from chemical damage. Now, even better, this information can be turned into chemical action that can do work through the proteins that are encoded by the DNA, not the DNA itself. And for me, this is a wonderful characteristic of life, all built on carbon-based polymers. The ability to encode information in the one-dimensional structures you see here, and these encode the proteins which can carry out chemical and physical work in 3-dimensional structures. It is a beautiful idea. Now, the second example of information in life I want to tell you about is all around regulation. That is, making the complex systems in living organisms act as a whole with apparent purpose. Now, before I talk about regulation in living things, I'm going to talk about a man-made machine, simply because it's easier to understand. And the example I'm going to talk about is a governor developed by James Watt to regulate steam engines. It was invented 100 years before that to regulate windmills. But this is a picture of a steam engine, or the governor from a steam engine, easy to understand. As this spindle rotates, as the engine goes faster, those balls swing out, shutting off the steam. And when they slow, they swing in and switch on steam. And this leads to what we call homeostasis, maintaining a constant speed of the steam engine. Now, the governor is an example of what we call a negative feedback loop, shown here in the context of a metabolic pathway in the cell in the upper of these two diagrams. The metabolic pathway goes from A to B to C, making different chemicals. As C builds up, it inhibits the enzyme catalyzing A to B, shuts off the synthesis of C, and that leads to homeostasis in the level of C. Negative feedback in cells was demonstrated brilliantly by the French geneticists Francois Jacob and Jacques Monod, who studied gene regulation of sugar metabolism in bacteria. Now, other control modules can lead to different outcomes, to positive feedback, because as C builds up, it accelerates A to B and makes more C, not less. And this module acts as an irreversible switch. Now, many such feedbacks and control modules operate in cells. They maintain homeostasis, they generate switches and timers and toggles and oscillators. They translate chemistry into informational modules. And these can be linked together to generate even more elaborate routines of informational management and processing. A metaphor for this is an electronic circuit of linked modules, as shown here. Now, although this metaphor is useful, a better metaphor for living systems, which I first read about with Dennis Bray, is wetware rather than hardware, because components in a cell can be relinked through solution chemistry. So it changes the hardware, if you like. And that, of course, increases the versatility of these control modules. Now, the management of life's chemistries by information allows the behavior of the complex system to act as a whole with apparent overall purpose. A purpose such as reproducing a cell, making a butterfly, or maintaining ourselves. Now, for centuries, the generation of apparent purposeful behaviors was thought to require a designer, usually a divine creator. And it took evolution by natural selection to change this way of thinking And that is my final idea: evolution by natural selection, how living things come about. Now, this is another beautiful idea in biology, mainly due to Charles Darwin. It has two parts. The first is that life evolves over time, and the second is a major mechanism for evolution is natural selection. That life evolves was actually widely discussed in the century before Charles Darwin, for example, by Jean-Baptiste Lamarck, and Charles's own grandfather, Erasmus Darwin, shown here on this picture to the left. Now, Erasmus was a colorful character. He was a scientist, a doctor, a poet. His research was written in poems. I have a number of first editions of his books. It's all written in blank verse. He was a Republican in the time of George III, refused to be George III's doctor, was a proponent of women's education and a great conversationalist despite a terrible stammer. He also argued for the evolution of life, suggesting simple, formulous mollusks could evolve into complex animals. Now, the evolution of life implies that there is a tree of life, that living organisms are related. If life only arose and survived once, as seems likely, given the conserved chemistry and coding, then all life on Earth is related to each other. And this connection of life was captured in the only diagram in Darwin's Origin of Species, published in 1859, where he illustrated this tree of life. Now, Charles Darwin gathered evidence in favor of his idea of evolution through natural selection during his trip around the world in the ship, the Beagle. Now, natural selection is a consequence of populations of a living thing, of a species, exhibiting variations which are caused by inheritable genetic changes. These can be caused by mistakes when the DNA is copied, or if DNA is damaged by external causes such as radiation. If these variants influence characteristics that make these organisms more successful, better adapted to their lifestyle and their environment, and therefore reproduce more, then that particular genetic variation will spread through the population. And over time, such genetic variations will accumulate and lead to changes in the population that the individuals of that new population will no longer be able to breed with the original population, leading to a new species, which is better adapted and more successful in survival and reproduction. Now, Darwin came to this view in part by studies of finches on the Galapagos Islands, although it was only when he got back in London and looked at them that he came to these ideas. Some of the finches shown here on the left that he described had big beaks that could break nuts, others thin beaks that could more easily catch insects under the bark of trees. They became adapted to their lifestyle and their environment by being specialized better for what they had to do. Just like different pliers shown on the right-hand side here are specialized to do different specialized jobs. But the difference between beaks and pliers is that pliers are intelligently designed by human beings for different specialized purposes, whilst the finch's beaks achieve specialized purposes by natural selection, without a divine designer or a creator. Now, for natural selection to take place, three things are needed. Firstly, reproduction. Secondly, a hereditary system. Thirdly, that hereditary system needs to exhibit variability upon which selection can work. Let me show how this works based on the ideas of the cell and the gene that we've been talking about. Imagine a single-celled life form with a brown cell coat, like the one at the top here, the color of which is determined by a gene in its chromosomes. Imagine when the cell divides and copies its DNA, it makes a mistake. So the cell coat gene is altered and the cell now makes a red coat, as you see at the bottom right-hand corner. Also imagine that red-coated cells survive better. Perhaps they do not taste good to a predator. And as a consequence, the red cells replace the brown cells in the population and evolution by natural selection has taken place. This is a profoundly important idea in biology, so much so that the 20th century geneticist Hermann Muller, who worked in the United States, used it to define what life is. He defined it simply: living things have properties which allow them to undergo natural selection and therefore evolve. I'm going to consider this a little more in a moment, but before doing that, I want to say goodbye to Darwin as an old man with the final monumental sentence finishing his Origin of Species. Whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Note how Darwin relates his work to the great physicist Isaac Newton. He was arguing that biology can have fundamental laws just like physics. Now, to help think about defining life, I first want to consider the difficult category of intermediate life forms, particularly viruses, which are, of course, of such interest to us today. And this will help, I hope, how we should think about the nature of life. Now, this is a virus. It's a virus of bacteria, bacteriophage. They have a genome based on DNA or RNA. Coronavirus has an RNA one, by the way. They undergo evolution by natural selection, and thus they passed Muller's test, but can only reproduce, only have a metabolism, essentially, when they are inside the cells of other host living organisms, and they do so by hijacking that cell's molecular machinery to copy the virus genome and to make more viruses. And this means that a virus is completely dependent upon another living entity. Perhaps you could say that a virus is alive when it's in a cell and a lifeless chemical when outside a cell. Bit of a cop-out, though. So the question is, are they really alive or not? Now, in thinking about this, it's important to remember that all life forms are usually to a greater or lesser extent dependent upon other living beings. The many parasites that live on or inside cells or bodies of animals, plants, or fungi are clear examples of this, although the extent of their dependencies are less than it is for a virus. There are certain amino acids that we humans cannot make. We get them from other living things. Even free living microbes are dependent upon molecules usually made by other living organisms, even if they are quite simple. Therefore, there's a graded spectrum of living organisms, from the viruses through to plants, with a wide range in between. And these different life forms have varied dependencies on other life forms. And in the case of the virus, that's very strong. In others, it's weak. But I will argue that these different life forms are all alive because they evolve by natural selection. And it means that life on Earth is fundamentally connected, both through being related as a result of evolution and also through these deep relationships and interdependencies. Now, I'm coming to the end of my talk, and I began it by saying, by considering the important ideas of biology, and I've considered 5, we could establish some principles to understand what life is. And let me now summarize where we've got to, though I should warn you, it doesn't lead to a neat dictionary-like definition of life. The first slide here identifies that living things are independent, bounded physical entities. They're not like a computer game. They're separated from their environment. They're in communication with their environment. And this allows them to generate order and organization. And the basic unit of life, at least on Earth, is the cell. The central principle for defining life is this one, the ability to evolve by natural selection developed by Muller. And by doing so, they acquire purpose. They can grow. They can reproduce. They are entities that have a hereditary system which exhibits variability, and that is the basis of evolution by natural selection. Living things are chemical, physical, and informational machines, as I've argued. They construct their own metabolism, which is used to maintain themselves, grow, and reproduce. And the final point concerns life on Earth, which is based on carbon polymer chemistry. These polymers produce nucleic acid-based linear information storage devices and protein-based chemical and physical machines. Integration of all these functions requires, of course, the management of information. Inputs are gathered from within and outside a cell, process, stored, and used to instruct cells and organisms to maintain themselves, to grow, and to reproduce. And living things on Earth are related by common descent and are closely interconnected and dependent upon living things. I warned you, it wasn't a simple dictionary definition. However, should we ever encounter life elsewhere in the universe, I suspect it will be based on rather similar fundamental principles. The details may be different. It could involve different chemistries. Perhaps chemistry is not based on carbon. However, for it to readily serve the need for long-term information storage, it's likely, I think, the chemistry will also be based on polymers. These life forms will need energy sources to power the processes of life. It could come from the nuclear reactions within stars that show light and heat, or from geothermal energy within the planets. But let's just finish by returning to our planet, the only corner of the universe where we know for certain life exists. The life that we are part of here on Earth is extraordinary, constantly surprises us, but in spite of its bewildering diversity, scientists make sense of it. And that understanding makes a fundamental contribution to our culture, to our civilization. Our growing understanding of what life is has great potential to improve the lot of humankind. But this knowledge goes further. Biology shows us that all living things we know of are related and they interact closely. We are bound by a deep connectedness to all other life, to infecting bacteria, fermenting yeast, to pea plants, and to flitting yellow butterflies, all of which have accompanied us through this talk. Together, all these species are life's great survivors, the latest descendants of a single, immeasurably vast family lineage that stretches back through an unbroken chain of cell divisions into the far reaches of deep time. As far as we know, we're the only life forms who can see this deep connectivity and reflect on what it means. That, in my view, that gives us human beings a special responsibility for life on this planet, made up as it is by our relatives, some close, some more distant. We need to care about it. We need to care for it. And to do that, we need to understand it. And that is why I've written this book. And it's also why I dedicated it to my grandchildren, who will have to do exactly that. Thank you very much for listening to me. It's been a pleasure to talk to you all this afternoon.
Hi, and I'm back, ready to ask some questions on behalf of the audience. We've gotten quite a few, and so many that sort of range from specific questions about the mechanics of evolution to questions about ethics. So I think I'm going to begin with this interesting question. We talked a little bit about technology and the ways that technology sort of forms a lot of our metaphors for thinking about dynamics. So I'm going to ask this question that's sort of looking towards the future and the ways in which the premise of transhumanism proposes the linking of silicon-based intelligence and carbon-based intelligence. And I guess the question is sort of venturing is, could we imagine the definition of life in the future consisting of things beyond carbon-based intelligence?
Well, we can, of course, imagine anything. That's the beauty of our imagination. And that we can extend our phenotype. That's a term used by Richard Dawkins in one of his books. We extend our phenotype in simple ways with tools and more complex tools. And so that's part of the way we are. We use our brains to construct tools that are useful to us. And you can imagine connecting it to more sophisticated tools, which are digitally based. We already do that in part by working through computers. I'm not sure it really undermines the principles that I've been talking about, because it enhances the basic processes, but doesn't actually substitute for the processes that are central to life. So yes, it's important to think about it. Yes, it's important to know where the future may take us. But in thinking about the definition of life, I'm not so sure it makes such a big difference.
Great, thank you. That's very clarifying. We have another question that's sort of linked to life, and that death is linked to life. But so Ana Maria Caballero asks, what is death then? Chemicals on their own are inert. What is the line between life and death?
Well, I was about to say I like thinking about death. Actually, I don't like thinking about death, but I think death is very illuminating, as the questioner says, in two ways, actually. The first way is that without death, actually, it's difficult to have life. And this sounds like a conundrum, but to have efficient and effective evolution by natural selection, you have to have successive generations. Otherwise, everything gets gummed up with the old versions that are on the planet. And so death advances evolution by natural selection. So that's one meaning. It's not the meaning the questioner had, but I think it's an interesting way to think about it. The questioner asks a very interesting question, and I'm not really quite sure what is death. I mean, it's obviously, in a sense, the breakdown in the ability of life to behave as a whole, and therefore to behave as with a purpose. So I think it's actually closely connected with the destruction of proper information management. So you may still have individual chemical reactions going on. I mean, of course, we know they can go out on outside a cell inside a test tube, but these are not linked together in an interdependent way that generates the overall purpose, like a cell behaving, homeostasis, reproduction, and the like. If we want to get more colorful and talk about the spark of life, for example, I sometimes think that the processes that produce energy, which is to do with the transfer of protons across membrane gradients, when that starts to break down and cellular life, and therefore life as a whole, cannot make the energy to maintain itself, in a sense, the spark, because it's electrical, essentially, it leaves the living thing, and maybe it's the death of that spark and the lack of energy that eventually leads to it not functioning, and therefore death. That was a long way of saying I don't know the answer.
Well, the questions are just as illuminating, and I think everyone will sort of benefit from that answer. So we unfortunately, I think, only have time for about 2 more questions. Sorry, everyone. Won't be able to get through everything, but I'm interested, and I think everyone else is interested. We've gotten a couple questions about this, the source of this spark of life. So I'll read this question from Frederick Gardy, who asked, in modern society with more variation in belief systems than ever, how can we develop a shared understanding of the origin of life and the spark that gave the first organisms the purpose of living?
Oh, well, I think I need a book on philosophy here at the very least, and probably theology as well. Within this question is the words, the origin of life. And you will notice I avoided that in my talk, and I barely touch upon it in the book either, because although I repeat the generally accepted view of how life might have arisen, and actually you have in Boston Jack Shostak, a friend of mine, who has thought a great deal of this, and it is the idea that life somehow began with RNA, because RNA can both encode information and also has sufficient chemical complexity to catalyze reactions. So if we're thinking about the origin of life, maybe we have to think about that type of solution. But it is, I'm afraid, pure speculation. And now when we come to different belief systems, which sometimes have speculated over the centuries about the origin of life, there are a wide variety of those. I'm just going to make it even more mysterious, because you can see I'm dodging the question. The amazing thing is this. You know, the Earth was formed about 4.5 billion years ago. It took 500 million years probably to cool down enough to imagine life. So we get down to about 4 billion years ago. And within 500 million years, we have what we think a single-celled life in fossils. And that is a remarkably short time when you think it took from 3.5 billion years ago to 500 million years ago. It's another 3 billion to go from a single cell to a multicellular organism of the sort that was found in the early Cambrian geological time. And this is extraordinary for me. How could a cell be formed in a few 100 million years? And yet it took 3 billion years to go from a single cell to multicellular life. There are still mysteries out there, and that's one of the good reasons for thinking and studying science, to see if we can make sense of them. It's not only useful, it's important for our culture and important for our civilization.
Amazing. So I think on the topic of mystery, since it sounds like that's a lot of what we've been discussing, I'll sort of end with this question from an anonymous attendee who just wants to say, along with everyone else, that this has been a beautiful talk, and thank you for joining us today. What would you say is the biggest mystery in human biology today, they ask?
Well, I think I just said what I think is the biggest mystery, at least in biology, of which humans are part, and that is our origins. I do not know how the origin of life came about. I can just about cope with the origin of humanity with our incredibly complex brain, but the origin of first life is the biggest mystery for me.
Great. Well, I think we can end there with the desire for more information and to find things ourselves and to read this amazing book, which I have put in the chat for all of you in a couple different places. Thank you once again to Paul for this fantastic presentation and to Andrew for your introduction. Thanks to all of you out there for spending part of your afternoon with us. Please learn more about this fascinating book and purchase What is Life at harvard.com. On behalf of Harvard Bookstore, the Harvard Division of Science, and the Harvard Library, all here in Cambridge, Massachusetts. Have a good afternoon, everyone. Keep reading and be well. Thank you so much, Paul, for joining us.
Thank you very much. Very much enjoyed it. Thank you to you both and all the audience.
Enjoy the rest of your day.